University actually is implement the outcome-based learning. So every questions we set is based on learning outcome. You should know, all of you should know. Okay, so that's why we have some questions about hierarchical organization. That means the flow. That means system, organ, tissue. There's organ, tissue, and the cell. Okay. So we have this kind of organization to questions to test your understanding. Okay. So secondly, you will go through have different kinds of process and talk about and also have question about homeostasis. Okay, that means osmoregulations and hormone. Okay, homeostasis. Okay, and also essential for survival. And also some of the questions will ask you why this kind of organ have a special structure to provide a specific function. That means assess the correlations between anatomical structure and physiological functions. Just like one you observe in the first assignment. Okay, I asked you why we have two types of mesophyll cell. Do you remember this one? Okay, because Perry say mesophyll, they pack. All of you have get correct. They pack on the top of the leaf so that they can absorb more light. And then the remaining spongy mesophyll, they are in granules because they need airspace for gases exchange. So this kind of question, okay, this is the style, okay. So and then one more is you need to know something different and something similar between plant and animal. Okay, I will have question about it. And then as some of them, I just give you some data. So for quiz, do you remember we have an MCQ showing the blood pressure and blood flow? This kind of action, this kind of question, because I want to achieve this learning outcome. Based on the graph, you already know which organ is it, which part of vessel is it. You remember that MCQ. Okay? So final learning outcome is the most difficult one. Integrating multiple processes together. So in after quiz, the system we teach usually including hormone system, okay, the endocrine system, okay, the hormonal control. Okay, so usually it's endocrine system with another system combined together. Okay, so and give out one function. Okay, so the question will integrate multiple systems, not only just test you digestive system, for example. Okay, not in this way. Okay. So for plant, so the hierarchical organization mainly focus on leaf, but for plant tissue, I remind you here. Okay, so there's three kind of tissue in plant. The outermost one called dermal tissue, not epithelial tissue. Not epithelial tissue. Epithelial is from animal. Do you remember? Okay, the names are the difference. Okay, so for transportations of material, that means put the purple color here. Okay, purple color here. So they are called vascular tissue, and yellow one called ground tissue. Three type of tissue. There's no four. No, there's only three. Okay, and leaf. Go to leaf, okay? Leaf we quite talk in details here. Leaf here. Okay, starting from here. Okay, so for dermal tissue we have cuticle. Okay, we have cuticle. Okay, and epidermis. Okay, leaf epidermis. Okay. And leaf epidermis and this vesicuticle also work in osmoregulations of plant, so reduce water loss. Okay, water loss. And also on the dermal tissues, it also include stomata. Okay, to control the opening of stomata, there's a pair of gut cells. Okay, pair of gut cells. And to control the osmotic regulation, vesicle and the stomata play an important role here. Bear in mind. Okay? So for ground tissue of leaf, okay, ground tissue, everyone knows. For maximal light absorption, there's some palisade mesophyll cell. And for another side of leaf, okay, there's some spongy mesophyll, making some airspace for gases exchange. Okay? And they are usually a parenchyma cell, metabolic active cell for photosynthesis. Okay? For photosynthesis. So for vascular tissues of plant, okay, vascular tissues of plant, they got vascular bundles, okay, vascular bundles, okay. But for there's a part, there's some cell, actually they are from ground tissue, which is bundle sheath cell. So bundle sheath cell, you see yellow in color, they are ground tissue, ground tissue, okay. For xylem and phloem, easy, okay. So they are in vascular tissues, okay, vascular tissues, okay. And on the leaf surface, they have some vessel we call wings, okay. It include both xylem and phloem, so they are vascular tissue, purple in color, okay. So how about animal? So for growth, I don't have any contents in the exam, so don't care about growth. No need to care, okay? So this is all about plant. So now we already in revision, okay? So just check the recording, okay? And then how about plant number 2, plant this one. Animals. Yeah, body plan. So for animal, so I still need to have some questions about hierarchical organization. But the focus, you can focus on epithelial tissues. Epithelial tissues. So in animal tissue, it divides in epithelial, connective, muscle, and nervous tissue. Okay, nervous tissue, 4 types. But this one only found in animal. The one in plant called dermal tissue. 1秒死咗。Okay, the outermost tissue of a plant is called dermal tissue. The tissue name, epidermis is organ name already. That's another level. Okay, okay. So okay, so in epithelial tissues, okay, so there's different type. Okay, different types. Okay, squamous, cuboidal, columnar. Okay, so they can be just simple, just that means one cell fake, and they can be stratified. Stratified, that means multiple cell layer. Okay, but some later you will see one of them, they are pseudo stratified. Okay, pseudo stratified. But in epithelial tissue, what you need to know is we have different kind of epithelium. But where can you find it? The target, the focus is where you can find it. Okay, so for cuboidal epithelial, you can find in kidney tubules, the nephrons. You remember the nephrons, okay? And for simple columnar epithelial, you can find in intestine, because one cell fake can help in uptaking nutrients or secreting liquid or enzyme for digestion. Okay. And for simple squamous epithelium, okay, they can be found in blood vessels and air sac of lung, okay, because they need to exchange material. For stratify, okay, want to hide. So for stratified squamous epithelia, because this usually can be found in skin and the lining of your mouth. Because this kind of surface usually they will have abrasions, this kind of thing. Some of the cells they will snap off during the process. Okay, abrasions. Okay. So another last one is pseudo stratified. They are not really stratified. They actually one cell thing. But they usually this kind of they are very variable in cell height. Okay, some cell they are taller, some cell they are small. But the most distinct characteristic is they are ciliated cell. They got some cilia on their cells, so that they can remove the dust together with the mucus. Okay, out of the respiratory tract. Okay, so this is also a kind of barrier defense in the immune system. Okay, barrier defense. Okay, so next one, nutrition. 跟唔跟到啊？ Can you catch up? Okay, so nutrition. Nutrition may have some contents in plant only. No, there's no content about animal. Okay, because we already test you. Okay, in the quiz. Okay, but the focus is photosynthesis and Kelvin cycle. But I want to tell you is that you need to focus on the graph. How the process occurs. You draw when you do when you do having revision. Draw the figures. Understand what happened. How the electron transfer between two photosystems. Okay, so there's two photosystems. For photosynthesis always starting the light absorption always starting from photosystem 2. Okay, photosystem 2 and then photosystem 1. This naming is due to their discovery. People firstly discover photosystem 1 and then photosystem 2. But for absorbing light energy and excitation of electron, it is starting from photosystem 2, and then the energy will transport to photosystem 1. Okay, the energy will transport I should say it this way, the energy from the photosystem 2 is for making ATP. So the electron will lose some energy and then enter photosystem 1 and absorb light energy again. Okay, this line. Okay, and this energy is to making NADPH. Okay, making NADPH for Kelvin cycle, carbon fixation. Okay. Okay, here, this one. Okay, so the graph is important. Okay, practice at home. Okay, draw it, understand what happened. Okay, for Kelvin cycle here, we obtain ATP and NADPH. They are from photosynthesis. Okay, so in Kelvin cycle, it is divided into three phases. Phase 1, carbon fixation. Okay, this graph is starting from 3 carbon dioxide molecules. Bear in mind, 3 carbon dioxide molecules. Okay, so and this after these 3 molecules enter the pathway, so each of the carbon dioxide will bind to 1 free carbon sugar. Okay, we call RUBP, ribose-based phosphate. Ribose-based phosphate. Okay, and then through the rubisco, it will convert into free carbon sugar form. 6 free carbon sugar. Okay, which is free phosphoglycerate. Okay, and then consuming ATP also, and then enter the second phase. Okay, second phase. So in phase two we call carbon dioxide reduction. Okay, you can just write reduction. I still give you mark. Okay, so now we will consume, this process will consume the ATP and NADPH from photosynthesis. Okay, and then making 6 free carbon sugar. Okay, 6 free carbon sugar, which is G3P. Okay, G3P. Okay, guys, well, LTI 3 phosphate, G3P, turn into this one. And 1 G3P will leave this cycle. And then remaining 3 G3P will enter phase 3 for regeneration. Okay, making 5 carbon sugar. Go back to, so this 5 carbon sugar is this one, RUBP. And then go back to phase 1. Absorb 3 new carbon dioxide again. So bear in mind for Calvin cycle, to make one sugar, you need to go through this cycle twice. Okay, so this word important. Go through 2 times, this word important. Go through 2 times, you will got 2 GVP. Okay, 2 GVP. 2 GVP we can form 1 glucose. Because glucose contains 6 carbon. 3 carbons cannot make it. So we need 2 free carbons molecule to make 1 glucose. Okay, this is the revision focus. What is next? Okay, circulation and transport. For plant, I will not show you the lecture notes. I already tell you. Okay, so for transportation, there's no content about in the exam. There's no content, but in circulatory and transport in plant, I already tell you how the plant perform osmoregulations. Okay, so it is by vesicuticle. Okay, so these vesicuticle we can reduce water loss, and opening and closing of stomata can control the water level of the plant. This is the direct way. Okay, the direct way to control the water level. Okay, so for animal is, so for focus, please take a look about the single and double circulations. Where is it? Okay, circulation here. So we have talked about open and closed, but we don't have any content about it anymore because you have already tested in quiz. Okay, so open, not open, single and double circulations here. Okay, so for fish, ray and shark, usually fish. Okay, so they usually have a single circulation system. Okay, single circulation system. That means only 1 pump. Okay, in their heart they only get 1 atrium, one atrium and 1 ventricle, and then pump through the two parts. Okay, first part they go through gill capillary, obtain some gases, and then go to the body part, provide gases. That means oxygens to the target tissues, and then they will go back to the heart. Okay, single pump. Okay, single pump. But for frog, okay, for amphibian frog, okay, so they starting from this level, and another vertebrate mammals, okay, they are no longer double circulation. They use when they are no longer single circulation, they use double circulations. Okay, so the characteristic, you can take a look of their heart. Okay, so inside their heart they have they have two pumps, okay? But for the amphibian, in the ventricle, they mix it together, okay? Two atria for frog, two atria, one ventricle, okay? And when the ventricle contract, the blood will pump out of the heart and then go through pulmonary cutaneous circuit. Okay, for mammals, pulmonary circuit. The name is different. Okay, because for amphibian, the respiratory organ also include the skin. Okay, so the name of the circuit is different. Pay attention to the name. Okay, pay attention to the name. So after go through the gases exchange, because double circulation, the blood will return to the heart and pump it out again. Okay, pump it out again and then enter the systematic capillary. That means for the muscle and for other organ. And same in mammals. But for mammals, in their heart, they got two ventricle. Two ventricle. Okay, pay attention. This one ventricle. Two ventricle. Circuit names are different. Okay, circuit names are different. Okay. Okay, so topic 4. So starting from topic 4, the content in the exam will be more. Okay, starting will be increasing. Okay, more content about exam starting from year 4 will be included in the paper. Okay. So topic 4 is talking about osmoregulations and homeostasis in animal. There's no content about plant. Plant already included here. Okay, plant is already here. So there's nothing about plant in topic 4. So let's check the notes. First one I want to talk about nephron functions. Okay. Think minutes on summer break? Okay, do you need a break? No need, okay? So for this lecture, okay, topic 4, okay, it is only about animals because plant we have vesicuticle and stomata to control the water loss, okay? So for animal, first thing you need to understand is the nephron functions. We see. Yes, starting from here. Okay, so for nephron, so the filtration step is the first step. Okay, the filtration is starting from glomerulus. You know, I think all of you know. Okay, glomerulus. Okay, starting from glomerulus, and then filtrate will enter the tubules. Okay, so and then each part of tubules, they have different functions. Okay, so for proximal tubule, this one is for primary reabsorption, first reabsorption. Okay, first reabsorptions. Okay, so and here is the material that will be reabsorbed in this part. Okay, I don't talk in detail, but you need to know the functions of each part. Okay, so say after filtrate pass through the proximal tubules, and perform some reabsorption, they will enter the filtrate will enter the loop of Helen. Okay, loop of Helen. So and loop of Helen will divide into descending part, descending. Okay, more descending, that means longer tubule, longer loop of Helen, more reabsorptions. Okay, more reabsorptions of water. Okay, and then it will enter the ascending. Okay, you revise by yourself. I don't talk in detail. Too many. Okay, too many. And pay attention for this So after leaving the loop of Helen, okay, they will enter the distilled tubule. So distilled tubule is responsible for the secondary, okay, the second reabsorption of nutrient. Pay attention, don't mix it up, okay? I will have some question to trap you, okay, here, okay? And after this part, all the filtrate will enter collecting ducts, okay? And then enter the urine pelvis to become urine, okay, okay? And then so between animals, okay, their net form structure, they are different, okay. So for some desert mammals, because they cannot obtain water easily, okay, they need to reserve water in their body. So in their kidney, most of them, they are justomedullary nephros. That means the nephron with longer loop of Helen to reabsorb more water, okay? Absorb more water. But for any mammals in moist habitat, okay, because they can drink water, they can access water easily. So for their kidney, usually only mostly are cortical nephron. So shorter loop of Helen, okay, So eventually their UV is more dilute, contain more water, because less water reabsorption in loop of Helen, shorter loop of Helen. Pay attention to it, okay? And also for exam, because one online outcome is integrating multiple biological process for functions. So you need to know osmoregulation in animal also is actually due to hormonal control, okay, the endocrine system. So we have 3 hormonal circuits here, okay. In assignment, we already have one. I remember should be ADH. ADH 我都唔記得咗出到自己 ADH. So I give you one way to have a revision already. Okay, you should know how they flow, how to control. Okay, which organ receive signal, give and produce some hormone, and then what is the effect of the hormone. That's why I want you to draw in the assignment. Draw the flow of the process. Okay. And there's one more, so other than ADH, another one is RAAS. So you please spend some time to revise all three of them. Another one, AMP, the last one is, remember, it's just opposite RASS, okay, this one, okay, opposite to this one, okay, so the flow is relatively simple, okay. So remember, spend some time to revise those content here. So next one is topic 5, right? Topic 5 is hormone. Okay. Hormone, what is it? Plant is actually including signaling. Plant here. So for plant, so nearly 50% of content is talking about plant hormone actually. Okay, 50%. Another 40 or 50% is talking about some signaling molecules, okay, or light receptors. Okay, for plant hormone, mainly I teach you 5. Okay, so for these 5 hormones, you need to know the functions of these 5 classic hormones. Okay, so just function only. Okay, what is the role there? Okay, what's the role there? For auxin, they trigger phototropism, cell elongations, and also some minor functions like plant development. Okay, so they will affect the apical dominancy. Okay, together with the next one. Okay, so auxin also affect the phyllotaxy. That means the leaf arrangement of the plant eventually. Okay, and also leaf patterns, the leaf wing patterns, and control the formations of female gametes, and control the growth of vascular cambium. Okay, because the secondary properties can stop. Okay, it's in special period. Okay, so this is controlled by auxin. Okay, so second type is cytokinins. So the most important things is they control cell division and differentiation. That means the role of the cell, okay, is controlled by the cytokinin. So if they are high in concentration of cytokinin, those cells will be in the wood, will proliferate. Okay, they will form some root there. Okay, so if they are in low concentration, they will forming some shoot tissues. Okay, so second thing is controlling the apical dominance. Okay, we will have questions about apical dominance in the assignment. But bear in mind, we have two scenarios. One is keeping it intact. You need to know both of them. Okay, one is keeping the apical bud intact. Another one is removing the apical bud. Okay, in last year we have short question about this part. Okay, so this year will not be short question. At least. Okay, so that means you can choose, and there may be questions in MCQ. Okay, no short questions. Okay, but for practice, you already have a written one in your assignment, so you have a chance already. Okay, so for cytokinin anti-aging effects. No. Okay, so gilberanin. So they have several functions. One is stem elongation, but this stem elongation is just blocking. Unlike normal primary growth, it's just blocking. Okay, it's a growth of four rows stalk. Okay, not a primary growth. Okay. Second functions of gilberanin is stimulate the fruit growth. Okay, so last year. Is it this one? No. Last year there's an MCQ, but I forget whether I have question about it this year. I forgot. But you need to know. Okay, gilberanin also stimulate the fruit growth. And one last function is germination. Okay, germination of seed. Okay. So ABA, next one, ABA. Okay, so ABA in comparison to other hormones, this one is the only one slow down the plant growth. So which means often antagonize the actions of growth promoting hormone. Okay, which is the three I've talked about. Okay, so for the functions of ABA, so first one is seed dominance, because seed they need to dehydrate so that they can survive for a long time, and ABA responsible to keeping it dehydrated. Okay, keeping it dehydrated and inhibit the germinations. Okay. So and second function is drought tolerance. So this hormone is linked up with osmoregulation in plant. Link up. Okay. So because osmoregulations is one of the organs is controlled by the the gas cell, I mean the gas cell controlling the opening and closing. And the opening and closing is due to the the amount of ABA there. Okay, ABA there. So you need to know, okay, what is it? It's more detail because they link up to other topics. Okay, more detail. Ethylene. Okay, ethylene. So ethylene, it induces a lot of response, but I only test the one. Only test you the one. Those not the one. Only test you those included in the lecture notes only. Okay. So for first one, it is triple response to the mechanical stress. Okay. So that means when the plant push again the obstacle, it stimulates the production of ethylene. And due to this high concentration of ethylene, the plant will slow down the stem elongation and then thicken the stem because they need to get ready to turn, to have a turn, okay? So now next step, grow with curvature, okay? And then so cause the stem grow horizontally. So when there's no more obstacle pushing on the plant, the amount of ethylene will decrease so that they will grow vertically once again. Okay? So bear in mind, triple respond to the mechanical stress. This is the only function found in high concentration of ethylene. So ethylene also responsible to senescence. Okay, senescence, that means program the cell death, the apoptosis for certain cells of organ. So for in the lecture notes, we use leaf as an example. Okay, so leaves they will fall in autumn. Okay, and this process is due to high level of ethylene. Okay, high level of ethylene. But bear in mind, this programmed cell death is not a bad thing, because through programmed cell death, plant can recycle the nutrients. Plant can recycle the nutrients back to their plant system and for other purpose or for reserve to tolerate dry season. Okay, so for ethylene, not only cause the senescence. Okay, senescence. Okay, it's also cause leaf abscissions, fall. Okay, the leaf fall. Okay, and one more functions about ethylene is fruit ripening. Okay, fruit ripening, because when the fruit develop from the flower, they are not mature and they are not tasty. Okay, for eating. And for fruit ripening, they need high concentration of ethylene. Okay, so high concentration of ethylene will convert the starch and the acid into the sugar. Okay, and then make the fruit become sweet. And of course, the cell wall are in mature stage, they are hard. So the second functions of ethylene in fruit ripening is breaking down the cell wall component, okay, and making the cell wall soften for animal to eat, okay, so that they can propagate the seed, okay, they can disperse the seed, okay. Okay, this is the functions of 5 classic hormone. And next one is phytochrome and its effects. Okay, phytochrome. So for phytochrome, there's a lot of functions. Okay, so any light event, okay, like any event that's triggered by light, except photosynthesis, we have a term called photomorphogenesis. And many of them, they are due to the photoreceptor called phytochrome. Okay? So for phytochrome, remember there's two form. these two forms is based on their wavelength they absorb. Okay, so let's go to next slide. So for phytochrome, their photoreceptor mostly absorb red light and far red light. So due to absorbing different kinds of light, so they return to two different kind of forms. Okay, so one form we call PR. PR stands for red absorbing form. Absorbing. Okay, that means it will absorb red light. Okay, another form is called far red absorbing form. PFR. Okay, important. This is important. Okay, I already have past paper. So if you are year 4 student, this is exactly the same you see in last year. This is past paper. Okay, in the assignment 2. Okay, past paper. Okay, so so for PR form and PFR form. So for PR form, it is, you can imagine it is an off switch, switch off. Okay. For PFR form, it is an on switch. Okay. This on switch will trigger different kind of response. Okay, different kind of response like seed germination. Okay. When PR form absorb red light, convert to PFR form. So this PFR form of phytochrome will trigger seed germination through signal transduction. Okay, and also it inhibits vertical growth and stimulating branching. Okay, and resetting internal clock, circadian rhythm. Okay, and one more thing is controlling the flowering. Okay, controlling the flowering. Okay, so I just want to highlight one thing because for seed germination, straightforward. If finally it is a PFR form, so it triggers the germination of seed, no problem. But for shade avoidance, okay, this process is rely on PR form. PR form. Why? Okay, let's go back. Okay. So PFR form on switch. Okay, take a look of the response. The response is inhibiting the vertical growth. That means keep it short, keep the part short. Okay, but now we are talking about shade avoidance. Okay, we want to absorb more light. That means plant growth taller. Okay, so it's opposite. For this process, shade avoidance, we need PR form. Pay attention, we need PR form. We need it switch off, stimulate the vertical growth, but limiting the branching. Absorb more light energy first. Otherwise, how can you get enough nutrient for branching? Okay, for branching. Okay, bear in mind. Okay, favor of PR. Okay, I already remind you here. Okay, circadian rhythm straightforward. If there's sunlight, okay, in the morning, okay, most of the phytochrome will turn to PFR form and then reset the biological clock. Resetting the biological clock. Still straightforward as seed germination. Okay, so no question about it. Okay, so remember. So for flowering, okay, so practice with the assignment. Practice it with the assignment. So I can tell you in exam paper because it cannot be the same as last year. So this time I will ask you both long day plant, the response of long day plant and short day plant, both of them. But in assignment, I remember should be one of them, but I forgot. Short day and long day, I forgot. Properly, long day plant. I just, yes, apple tree. Yes, apple tree. So apple tree is a long day plant, actually. Long day plant. Okay. So now you need to know both of them. Okay. So first of all, for short day plant, I explain once again, because you need to understand, otherwise you cannot answer the questions. So why we call short day plant? Short day plants, because this is the photo period when you cultivate this plant. Okay, so for each plant they have a critical dark period. Critical dark period. Okay, so if the light period exceeds this critical dark period, so this is long day. We call long day. Okay, long day. Okay, so if it doesn't exceed the critical dark period, we call short day. Short day. Okay, so for short day plant, they will flower. Okay, so if you add one more flash of light to interrupt the dark period, so it will become a short night. So for long day plant, they have another name called long night plant. But when I teach, I usually keep it this word because you will be confused. But bear in mind, when you add a flash of light, you will disturb a long night, so it will become a short night. Okay. So it will become a short night. So short day plant, they will flower. Okay. So vice versa, long day plant, they only flower when the light period exceeds the critical dark period, and also when you interrupt the long dark period. Okay, because they have another name called short night plant. Okay, so at least you know. But for phytochrome, okay, so now we increase the difficulties. Okay, Phytochrome, we add phytochrome here. Phytochrome also have responsibility here. Okay, so usually I will put some red light and far red light flash here to interrupt the dark period. I will add it here. So in the assignment you can see I just randomly add the color of light there. Okay, so but no matter how I set, take a look of the last one. The critical one is the last one. Okay, so just like let's take last two examples. Okay, to teach you. Okay, so the last light flash is red in color. Okay, it's red in color. So when the phytochrome absorb red light, it will turn to yes, PFR4. So this is true interrupt of dark period. True interrupt of dark period. What does it mean? Long day plant will flower. Okay, because they have another name called short night plant. Okay, long day plant will flower. So if the last flash of light, it is in far red color, what is the form of phytochrome? Dominant. Which one? Yes, PR. Okay, PR off switch. Okay, so this is no longer an interrupt for the dark period. This is not an interrupt for the dark period. So long night. That means short day plant, flower. Got it? Important. Okay, this must be included in the exam. Okay, because you have an assignment to practice. Okay, how to answer the questions. Okay, so this is all about hormone for the plant, and then animal. Oops. 咦，原來已經11點幾啦，so I have to hurry. Okay, animal. Okay, so animal part, the endocrine system. Okay, water and lipid soluble hormone. Where is it? Okay. Okay, starting from here. Okay, starting from here. Okay, so for in animals, there's some water-soluble hormones and lipid-soluble hormone. Okay, hydrophilic and hydrophobic hormones. So you need to know the chemical structures. Chemical structures, what are they? What are they? Okay. So for water-soluble, usually they are peptides, polypeptides, like insulin. Okay. But for lipid-soluble, usually they are steroids. Okay, steroid. Okay, like cortisol. Okay, they are lipid-soluble. But for amine, some of them they are water-soluble. Some of them they are lipid-soluble. Okay, chemical structure you need to know. And then, so due to different solubility, they are these signal molecule, this kind of hormone, they are transported in different ways, different pathway. Okay, different pathway. So spend some time, take a look. Okay, spend some time, take a look, and revise. Okay, revise. So for water-soluble hormone, they are secreted by exocytosis and travel freely in the blood vessel because water-soluble. But for lipid, they need to they exit the cell, okay, by just diffusing out of the membrane. Membrane is made of phospholipid, light dissolved light. This is chemistry, okay, light dissolved light, okay, so that they can just exit, no need exocytosis, okay. But because they are lipid soluble, so in the circulatory system, they need transport protein to bind on it, make it soluble, okay, make it soluble in the bloodstream, okay. So and then when they reach the target cell for water soluble, they because of phospholipid membrane, they need receptor protein to bind on it, okay. But for how about lipid? They diffuse inside to the cell freely, okay, because of lipid soluble membrane, and then bind to the receptor in the cytoplasm or nucleus, okay. Different path you need to know. What's next? Tropic hormones, okay. Tropic hormones. So endocrine system, they usually not working alone. They actually controlling the response in other systems. So multiple systems they work together. So for tropic hormone, where is it? Tropic. Not just growth hormone. It is tropic and non-tropic. There are two kinds. Okay, let me find. Tropic hormones. I'm probably not teaching directly here, actually. But for tropic hormone and non-tropic hormone, you need to know the difference. Let's take growth hormone as an example. Growth hormone, okay, growth hormone. Okay, so tropic hormones, that means, so after anterior pituitary releasing this growth hormone, for tropic effects, okay, if you play a role in tropic hormone, okay, they will stimulate another organ to make the response, okay, okay. So for non-tropic hormone, they just make the response directly, okay. They will not involve any other organs, okay. Okay, thyroid. Okay, let's talk about thyroid first. So for thyroid regulations, so we have I teach you quite detailed, okay, and this is the tropic hormone, okay, this is tropic hormone because for thyroid hormones, it is starting from posterior in anterior pituitary. Okay, anterior pituitary. Okay. So they will have some thyroid releasing hormone from the neuron sensory cell. Okay, they release it. Okay, release it, and then causing the anterior pituitary to secrete thyroid stimulating hormone. Okay, the thyrotropic hormone, and this tropic hormone after transporting through the circulatory system, they will reach the thyroid glands, organ on your neck, inside neck here. Okay, so and then stimulate the productions of and the secretions of thyroid hormone. Okay, so this part you need to study in detail. Okay, study in detail. Okay, detail how the hormones interact with each other and where does it from? Where does it from? Because we have different hormones here already. One is from the brain, one is from the anterior pituitary, and one more thyroid glands. Okay, so and then what is the function? What is the functions? Okay, what is the function? How they form a negative feedback loop. Okay, because high level of thyroid hormone eventually will inhibit the secretions of thyrotropic releasing hormone, or TRH, negative feedback loop. Okay, to go back. So and for thyroid hormones, there's two types, and you also need to know what happens if people cannot make enough thyroid hormone. So why people cannot make enough thyroid hormone? Because they are under iodine deficiency. Okay, deficiency. And also in other animals, thyroid hormones, they have other functions, like frog here. This hormone contribute to the metamorphosis. Okay, so this is content covered in topic 1 already. Okay, but thyroid hormone play a role here to stimulate the reabsorptions of tadpole tails. Okay, and turn to frog. Okay. Last one is okay at the end. Okay, so it's the hormone in the adrenal glands. Okay, adrenal glands, there's two regions producing different kinds of hormone. Okay, two regions. One is medulla, another one is cortex. Another one is cortex. Okay, so in medulla you will find adrenaline. Okay, adrenaline. Okay, that means epinephrine and norepinephrine. Okay, this is they can be neurotransmitter and hormone, but we just only talk about how they work when we're working as a hormone. Okay, this is the function you need to know and the effect you need to know. So for cortex, there's another two types of hormones. Okay, so which is glycocorticoid, okay, corticoid, and mineralocorticoid. Okay, so the example is cortisol and adenosterol. Okay, so you also need to know their function. Function is here. Okay, spend some time and then revise the notes. Okay, so topic 6. One more hormone. So in here I have prepared one more slide, comparing two kinds of hormone. So and here is the difference. So at least you need to know some of them. If you don't have time, you don't need to memorize all of them, but you need to know some of them. Otherwise, you write nothing in the exam paper. You write nothing here. The easiest one is modulation by nervous system. easy. Plant do not have nervous system. Okay, this is already one of the difference between plant and animal hormone. Okay, so this plant hormone is not motivated by nervous system. Plant do not have it. But plant, some of the organ can control it by impulse actually, but I didn't teach you. Okay, there's too many here. Okay, so important things here. Topic 6, reproductions. For reproductions, let's talk about plant. Okay, I already simplify a lot for plant. Plant are difficult. So first of all, angiosperm should be 5 derived trait of sick plant in evolutions. No worry, I will disclose this recording in OLE, but you just spend some time to take a look. Okay, so bear in mind, from plant reproductions, we have some slide due to their evolutions, talking about their evolution, but don't mix up with the sick plant because they are all 5 of them. Okay, this one is you need to pay attention, not this one. This is talking about how the plant derived trait from evolution, why plant become dominant organism in the nature. This is talking about this one, okay? But for seed plant, it's talking about these 5 traits. Okay, I don't talk about it in detail because actually if you study angiosperm life cycle, it's covered all of them already. Okay, cover all of them already. So next one is for angiosperm. Okay, flowering plant. So they got flowers, and it is one of the key traits for their life cycles. Okay, flowers. Pay attention to the organ names. So for a complete flower, it must have 4 this type of organ: carpel, segment, stamen, petal, sepal. These 4. You need to know their name. Okay, remember, don't write anther, filament. This is smaller part of stamen. Okay. Fruit no need, germination no need. So, angiosperm life cycle. So because I only have 10 marks for long questions, so I will not let you to write the whole cycle. If I can prepare a question which can include 20 marks, I will let you to write, but I cannot. So don't worry, okay? We will not have written questions. So you see what I mean, okay? For angiosperm life cycle, okay? But I will be focused on numbers, the numbers in the life cycles, because we will have a lot of numbers here, okay? So like embryo sac formation, one megasporocyte eventually will form 4 haploid megaspore. Okay, lots of number here, so you need to pay attention to the number. Okay, for a complete embryo sac, that means a mature embryo sac. So pay attention, it should be 7 cells with 8 nucleus. So we have MCQ in tutorial. Practice, it will help. Okay? It will help. Just try to change some number when you practice. Okay? Change the answer and then you will understand. Okay? So why we got 8 nuclei? So 2 from synergic cell. Okay? The door guard, I would say. Door guard, okay? The door guard. Okay, so these are the cells attracting the tube cell, the forming the pollen tube. Okay, to the embryo. Okay, so that's why we have synergic cell. So in the other side, there's 3 more cells called antiproto cell. No function right now. We don't know the function right now. Okay, but at least we know 3 cells. Now we got 5. 2 plus 3, 5. Okay, 2 others, 2 and 1 cell. Okay, 1 cell called central cell. Central cell is special. They got 2 nuclei. 2 nuclei. Okay, 2 polar nuclei. Okay, 2 polar nuclei. And then there's 1 more egg cell just near the synaptic cell. Egg cell. So total 7 cells, 8 nucleus. Because one more nucleus is from central cell. Okay. So and this is also about the male one. Okay. The male the sporangia. Okay. At each alpha they got 4 microsporangia. 4 microsporangia. So if say micro, it will make a lot of dipole cell. That means microsporocyte at the beginning. At the beginning. After meiosis, it will become haploid. Okay, it will become haploid. So, but inside one pollen grains, you will got 1 generative cell. But when the pollen grain mature, it is enclosed by tube cells. Because once they arrive, the stigma receptive surface, they are not sperm. So they need tubes to guide the generative cells to the embryo cell. Okay? So the tube cell forming pollen tubes. And then the generative cells, here, 1 more numbers. The generative cells, they will form into 2 sperm. Okay? Bear in mind 2 sperm here. 2 sperm, also number. Okay? Pay attention. So why 2 sperm? Because second key trait of angiosperm is double fertilization. So double fertilization means 1 sperm fertilize the egg, another one will fertilize with the central cell. Okay? So and then eventually the central cell will become triploid. Okay, 3N. Okay, 3N, triploid. Okay? And this central cell will give rise to endosperm, provide nutrients for the seed. Okay, provide nutrients for seed development. Okay, and of course for the egg is develop a new plant. Okay, so that's why double fertilization they got 2 functions here, 2 functions. Okay, bear in mind 2 functions. Okay, this is all about angiosperm life cycles. So and okay, the remaining content is one at the back. There's one more about asexual reproductions. So plant also can undergo asexual reproductions. Asexual reproduction, just making 1 clone, 1 more clone, same genome. Okay, same genome. Okay, replicate 1, same plant. Okay, the mechanism they use is I tell you, okay? They use it here. Okay, I already tell you. Okay? And then, so they have because fertilization, they make a lot of pollen grains. They disperse. Okay? But some of them they may accidentally fall into the same plant. So plant they have some mechanism to repel their own pollen grains. That means prevent self fertilizations. So in organ levels, so we have several ways. One is diospecies, species. Okay, that means they only have carpel or they only have stamen. Okay, so that they can not. So one flower disperse pollen grain, another flower receive pollen grain. Okay, so second way is controlling the maturations. Okay, sometimes alpha grow first. Okay, the alpha grow first, so the alpha will be taller. Okay, the stamen will be taller, and then the stigma. Okay, the stamen will be lower. Okay, it can be vice versa. It can be different. Okay, but this is another way. Okay, one taller, another one shorter. So the most common is using self-incompatibility. We have two ways. One is gametophytic. Okay, gametophytic. So if the plant repel the grain through gametophytic self-incompatibility, so you need to pay attention to the flower chromosome set. Okay, so that means take a look of this one. Okay, the flower set. So do you understand this way? Do you remember? 唔記得晒啦，記唔記得？唔記得。Okay, so I tell you one more time. Okay, so for gametophytic, because you need to understand. Okay, otherwise you cannot answer the questions. So for a plant or for a flower, usually they have a pair, two set of chromosomes because they are diploid. They are diploid. Fertilization, diploid. Okay, diploid plant. So inside theirs, they have some S and U. So every plant, okay, even one population, they have S and U. But S and U means just same region. But A plant and B plant, although they have same species, they are S gene content, their S allele content, they are different. Okay, their S allele content they will be different. Okay, so for plant, they which contain S1 and S2 gene there, they can make the pollen grains, which can be S1 or S2. Okay, so when these pollen grain reach the plateau of S1 and S3 plant, okay, because this is S1, it will repel by the plant because S1, this S1 gene is the same as the plant. Okay, but for S2, it can fertilize because S2 gene, the chromosomes, the S allele is not the same as the one you find in their chromosome. It can fertilize. Okay? Another type, sporophytic. Sporophytic self-incompatibility. Sporophytic self-incompatibility. Okay? So this one you need to understand. For the flower, for example, for the flower contain S1 and S2G, they can make pollen grains of S1 or S2G. But no matter which kind of pollen grain, they contain both S1 and S2 gene product on their pollen grains surface. So you can see this one S1, but on the surface they got S1 and S2. S1 and S2. Okay? So now for such cases, so when these pollen grain reach the receptor surface of S3 and S4 flower, they can fertilize because none of them are the same as the flower chromosome set. Now, can you catch up? Okay? Okay, understand? Okay? So if this S1 pollen grain reach to the flower containing S2 and S3 set, so S1, because S1 still have S2 gene product on the pollen grain surface. Do you see S2? This one, there's a word S2, and here S2. None of them. So it still cannot. So this S1 cannot fertilize. So there's no pollen tip. formed in this flower. Okay? So pay attention. So pay attention for sporophytic. So you need to also look at the parent plant. Okay, the parent plant. Okay. So no matter which kind of pollen grain they make, on their surface they got both gene product, both S allele gene product. Okay. Okay. Animal. Animal. Number 10. Okay, animal. Animal part, so first of all, asexual reproduction. They have several mechanisms. And pay attention this one. Oh, okay? Only this one share with plant. Okay? But they got 4 originally. They got 4. And then so another focus is gametogenesis. So in gametogenesis, we have talked about spermatogenesis. That means development and formations of sperm. Okay, so you need to know whole process. Another one is oogenesis. Okay. So you need to know process. I will not talk about it one by one anymore because spend a lot of time. And another focus is ovarian and uterine cycle. Okay. So based on this arrangement, female reproductive system is more important than the male. Okay, if you're running out of time, focus on female one first. Okay. Focus on female one, okay? So I don't talk into detail, it's too long, okay? So if you still cannot understand, go back to check and my lecture notes, or you just see the recording. And Simon, you asked me to post the whole figure. This is the old version this is the figure from old versions of reference book. So I already posted in OLE, everyone can access. Because the new version is already cut it in small pieces. It's more clear, so I use this one. But if you want to know the whole picture, there's one. Yeah, there's one in OLE. Okay. Okay, so for the rest, no need. Okay, for the rest, no need. Last chapter. Almost. I already help you to reduce 60% of lecture loads. You can understand, okay? You still fail, hopeless. I will say hopeless, okay? Okay, so plan. I just test you one thing. Already included in your assignment. If you don't know how to do it, oh my God, because you just copy. Don't copy it, okay? So you need to differentiate between, so plant in the immune system, they only have innate immune response, no adaptive, okay, innate response. So in the innate response, they got 2 types. One type is palm-triggered immunity. Palm-triggered immunity, why we call PAM? PAM full name is called pathogen associated molecular pattern. They recognize pathogen patterns. Okay, so one example is using bacterial flagellin. The protein is the flagellin. Okay, so they got top lyse receptor for recognition of such protein. Okay, so after recognition, of course, signal transduction inside the cell, and then trigger the immune response. The immune response here is secreting broad spectrum antimicrobial chemicals called glycoalexin. This is the consequence. In assignment, you should answer up to here. No worry, assignment is just drawing the pathway, okay, but not up to here, okay? So another height, easy. Effector trigger immunity, because sometimes this top line receptor is blocked by the pathogen. But pathogen, because they need to take the nutrients, get benefit from the plant cell for reproduction. To divide many bacterial cells, they need to survive in the environment. Okay, they need to proliferate. So they will secrete some pathogen proteins we call effectors to the plant cell. But the plant also have their mechanism. Okay, that means they got a lot of disease-resistant gene in their genome, and then making some disease-resistant protein. Okay, and the effector here will activate this R protein and trigger two defense response, more stronger response. Okay, one local one is hypersensitive response. Another one is calling help or just stay alert, telling the plant stay alert. Okay, general defense called systematic acquired resistance. What is it? Okay, hypersensitive response, not only producing phytoalexin, an enzyme to damage the pathogen, but also this hypersensitive response include the formations of lesion, forming a scar. Okay, a lignin made a scar. Okay, this one. Okay, making some scar to hinder the spreading of pathogen, because they don't have nutrient anymore. They stop their spreading. Okay, so second response. Okay, stay alert, calling the plant stay alert. Okay, so it's systematic acquired resistance. That means the plant will send a signal molecule, which is methyl salicylic acid. This is the signal molecule, come from the infected area, sent to the different parts of the plant through phloem, and when they reach the target region, they convert back to salicylic acid, and this is the one to induce the systematic acquired resistance. And this resistance is a biochemical change, non-specific. Okay, and this moment they will keep fight against the pathogen for a few days. Okay, just 8 pages, no more. Okay, no, not 8 pages, just 6 pages. Okay, last one. Okay, animal, the things we talked today. So for invertebrate, there's no content in the exam. Okay, so don't spend time on that. So the first thing you need to know is barrier defense. Barrier defense. Okay, so barrier defense is innate immunities of vertebrates. So one of them is the major one is the mucus secretion, the mucus membrane. It can be found in different kind of system. You see, digestive system, the stomach lining, the intestine lining, we got mucus. Okay, mucus. Okay, respiratory mucus. But remember, the epithelial tissue there is pseudo-stratified, okay, squamous epithelia. Okay, connect with each other. Okay, they got hair pushing dust together with mucus away from the respiratory tract. Okay, so another body secretion like lysosome in the saliva, they also destroy the pathogen cell wall and stomach. The acid there also kill the pathogen. I better use PDF file because I keep jumping in different pages. Between different pages. Where is it? Next one. Immune cell. Okay, immune cell in native and adaptive immunity. Okay, where is it? So for innate, starting from here. Okay, so in innate immunity of vertebrates, we got neutrophil, macrophage, mast cell. So mast cell is not doing any killing, but they are secreting histamine for inflammatory response, getting red, warm, and swollen surface. Okay. So in the innate system also have dendritic cell. This one also can engulf pathogen. You remember when we talk about adaptive starting point, okay? We need to detect the presence of pathogen, okay? And dendritic cell and macrophage doing the same thing, phagocytosis, okay? And eosinophil is responsible to defense against multicellular invaders. That means parasites, okay? And NK cell, natural killer cells. So natural killer cell is not targeting to pathogens or parasites, but they targeting the infected cells and tumor cells. Okay, so different things. So for how about adaptive? Adaptive, you got two, T cells and B cells. B cells responsible to secrete antibodies. Okay, T cell is to recognize and activate and recognize the pathogen invasions, okay, by binding to the receptor of other immune cell like macrophage and dendritic cell. Because when the phagocytosis occurs, they will show up some pathogen antigen fragment with MHC molecule. Okay, I have talked about it today. Class 2, okay, class 2. And the T cell can bind to this receptor and become activated. Okay, B cell, T cell, adaptive immunity. So what's next? Okay, innate inflammatory response. Here. Okay, so this is quite complicated, although it's just below 100 words, but this process already involve different kinds of process. Okay, so you need to know all of them. Okay, you need to understand and write it by yourself. Okay, whole process by yourself. Okay, so you can see not only innate immune cell, you can see macrophage, neutrophil there. There's some innate cell doing other things. You see mast cell secreting histamine to trigger the blood vessel dilation. More blood, okay, more neutrophil, okay, more neutrophil, and so that the skin surface will become redness, okay, and warm, okay. So and remember, pathogen here also activate complement system. This one, complement system, they will activate it, okay, and activate it. But I don't need you to write that follow the whole of the one I teach you today. I taught you today, making a hole on the pathogen surface and then the cell lines. No need. Okay, but you know here at least inflammatory response. Okay, it will activate the pathogen here will activate complement proteins and promote further release of histamine and attracting more phagocytic cells. Okay. Remaining, I think you can do it ourselves. Okay, because you spend time, take a look, and try to revise, no problem. Last one. 4 major characteristics of adaptive immunity. So not just 4 points, not just these 4 points. So you need to know. So student ask me question is correct because the first one is the most difficult one. First one is most difficult one because you need to know how to make different kind of receptor on the board, on B cell and T cell. They rely on gene recombination. Okay, gene recombinations. And for second characteristic, remember it is cell tolerance. In bone marrow and farmers, they have some cell have got some antigen receptor to test the cell reactivity. If they can bind, if the B cell or T cell they can bind to this antigen, they will undergo apoptosis or make it non-functional. Okay, so we call this self-tolerate, not eliminate. Okay, because not all of them they eliminate. Okay, so proliferation. So that means when they can detect, when they bind with the antigen, they will become activated and making a lot of identical cells. Okay, some of them will turn to memory. Some of them they will become effector cell. For B cell, it will be plasma cell, secreting antibodies. And for T cell, it will be memory T cell and cytotoxic T cell. For effectors, okay, it will be cytotoxic T cell and helper T cell. Okay, helper T cell. Last one. Although you have tests, but still spend some time, practice how to write, how to draw the figure. Also including the content of x-axis and y-axis. At least you give me some label there, so that I know what you're writing, not just writing draft a wave there and then assume I can understand your wave. Okay, you got it? Okay. So what is primary response? What is secondary response? Okay, it's all of them. The remaining, I think, is too difficult. For knowledge, I can teach you, but I think it's up to here. Okay? There's too many, because some of you maybe cannot catch up, so I better test you up to here. Okay? This is all about revision. Okay? So